Hello friends, today we are going to discuss about one of the important topic in developmental biology about embryonic induction and competence. It is in continuation to our previous classes. That means blastulation and gastrulation. Okay, embryonic induction and competence. Embryonic induction and competence. You know, in our previous classes we have discussed about blastula becomes gastrula by invagination, what we call in-pocketing of blastula. And this gastrula, the significant event of gastrulation is formation of three primary germinal layers. That means ectoderm, mesoderm and endoderm. What next? This gastrula, that means the primary germ layers, has to undergo differentiations. We are during gastrulation we have discussed about different morphogenetic movements like involution, which are responsible for different, that means gastrulation process and its subsequent process. Okay. And now the cells of different layers have to undergo differentiation. Differentiation means for what? So that they become differentiated into specific organs, say kidney, say limb, say lung, say liver, eye, like that. So that there will be a proper organogenesis. We already discussed gastrulation is a crux point for segmentation or the multicellularity. That means organogenesis here. For that, there should be a proper cell differentiation. How this cell differentiation will perform? Whether it's a homogeneous, that means each and every cell has their own mechanism? No. There are certain cells which influence, that means which induces this differentiation, so that some cells will differentiate into specific tissues or organs, what we call embryonic induction. That means, it means that with some cells in the embryo we undergo differentiation based on the induction of some other cells. It's like in bottom line, X influence Y so that it can become a particular organ, and A after it forms influence Z, that means induces Z so that it forms into B organ. That means like a sequential induction. So it's all about embryonic induction. Induction, it has to, that means one has, X has to induce Y and Y has to induce Z, likewise. Okay, and since here since the differentiation is dependent, it is considered as a dependent differentiation, otherwise called inductive interaction. It's all about interactions now. So embryonic induction is otherwise called dependent differentiation or inductive interaction. Dependent differentiation. Dependent differentiation, otherwise called inductive interaction. It's a tissue interaction. How they interact, and we have to learn about specific words under this embryonic induction before we go into the exact topic. There will be an inducer. It is clear that certain cells act as inducers, that means what we call inductors. Certain cells which are come under the influence of induction, and there are certain mediators to perform this phenomenon, what we call inductors here. Inductor. Cells which are inducing are inductors, and which are under the influence, they are known as competents. Competents. You must be knowing about the relationship between the two, embryonic induction and competence. That means inducing, induced. Here inducing are called inductors, under the influence of induction are known as competents. How? Evocators. Evocators. This is very important. That means there are chemical messengers, what we call stimulus, chemical stimulus they release, so that the competent shall undergo, that means shall undergo differentiation to become specific organ, say organ A or organ B, depending on the kind of interaction. What takes us, that means inductors through chemical stimulus, evocators on that means what we call receptive tissues, what we call competence, so that they can differentiate into specific organ types. These are about embryonic induction. Okay, so there are inductors, there are competents, there are evocators, so that they become into specific organ. And it is a sequential phenomenon, not just a single linear, it is a single step phenomenon. Sequential, and this specific organ become a source of induction. That means it again in turn acts as inductor. Influence, that means induct some other competent, say one, so that it become some other organ, say organ B. Likewise it will repeat it. That means in a sequential induction, we will discuss in this episode, for example, how it performs. It's about overview. So embryonic induction is a sequential process wherein some cells induces some other cells so as to differentiate into some specific organs through evocators. That means chemical stimulus. That's it. And but the most important thing, we have discussed morphogenetic movements. Here, they are the same result, morphogenetic effect. That means development into a specific structural pattern. Cell differentiation means, first of all, and where it influences at the molecular level, the influence of protein synthesis. That means accordingly the cell behave. That means this different cells, say kidney cells, liver cells, have different cell behavior. How? They have different protein synthesis mechanism. That means different molecular mechanism. So at this level they start so that they become differentiated into a specific organ type. That means what we call morphogenetic effect is the result of embryonic induction. It's all about the basics, and actually we all knew that about embryonic induction, but at the in previous days nobody knows until the remarkable what we call path-breaking experiments done by Hans Speeman and Mangold. They performed, that means they did experiments on neural induction in amphibians in 1924. You know, Hans Speeman and Mangold. In 1924, they did experiments on neural induction in amphibians. And later onwards, Speeman in 1935, he was awarded Nobel Prize for his path-breaking what we call organizer's effect on embryonic development. By using newt eggs. He didn't use frog eggs but newt eggs. I will tell you how, that means what makes him to use the newt eggs but not the frog eggs. So for here, Hans Speeman. Speeman is very famous embryologist, and actually he is a German geologist. Based on his remarkable experiments in embryology by using newt eggs, he has identified, he discovered the concept of organizers. I will tell you what exactly organizers and its effect on embryo development. And for his remarkable contribution, he was awarded Nobel Prize in 1935. And the first most primary, that means what we call a path-breaking pioneering experiment done in the same, that means Speeman and Mangold in 1925 with respect to neural induction in amphibians. From then, several experiments has been performed by different scientists that broaden the basket, what we call conceptualization of embryonic induction. It's all about different basics, okay? And before we go on topic, we have to learn about certain different experiments done by Speeman. For convenience, for understanding, there are we have discussed two particular experiments. Speeman experiments. In experiment 1 and experiment 2. For understanding how he devised the concept of embryonic induction. In experiment 1, we have discussed already, he has used the newt eggs but not the frog eggs. Generally, in amphibians, frog eggs are the most favorite objects, but he has used the newt eggs. Why? Because newt eggs are essential. That means they are convenient for surgeries. for transplantation experiments. At the same time, we already discussed animal pole and vegetable pole will be there in the two poles in an egg. Here the animal pole is colorless, whereas the vegetable pole is dark. That makes him to for easy differentiation is very important. So based on these two things, he has used the new text but not the frog eggs. And then coming back to the experiments, in one particular experiment, to understand the embryonic induction, that means how one particular cell influences others or one particular region. Say this is a new egg, okay? He has bifurcated by means of a baby egg through noose. Noose is nothing but a loop with a tightening knot, just like we tie up with a thread. Now, it's the same concept. Say here this is a new egg. This is a new egg. This is the animal pole, and this is the vegetable pole. Okay. We knew that here the nucleus is present in animal pole, but not the vegetable pole. And you know what he did? He has constricted the egg through this baby hair. Constricted the egg so that it can almost it appears like two eggs. But the thing is, constriction is through gray crescent. Gray crescent. What is gray? Gray crescent is nothing but an egg that is opposite to the sperm entry. And this gray crescent divides eggs into two, animal and vegetable. And this, what we call noose passes through this gray crescent, exactly dividing the egg into two. You know what happens next? You know what happens? Here, that part of the embryo which contains nucleus undergoes cleavage, but that part of the embryo, that egg which doesn't have nucleus, that means what we call enucleated, doesn't undergo any cleavage. It becomes passive. That means cleavage activity is reported in the egg, that means with nucleus part, embryo with nucleus, but in case of enucleated zone, there is no that means cleavage. It's what we call passive. Say for instance, for understanding, I will write like this, okay? Here, this is the egg partner, the two, but here it remains like passive. That means two blastomeres you can expect, but here just one. The same I am magnifying here, okay? It remains passive and remain active. Passive. But at 16 cell stage, but the thing is, the two part of the embryo are still connected by a narrow cytoplasmic bridge. They did not detach completely, but there is some kind of narrow cytoplasmic bridge. Through this cytoplasmic bridge, at 16 cell stage, the nuclear material from the nuclear part or portion has been transferred to the enucleated part. Say from here to here at 16 cell stage. At 16 cell stage. So that the enucleated part become nucleated part and become cleavage active. And ultimately, later on what he did, he has detached the two parts simply by tightening the knot, so that each part become a full-fledged embryo. That means full-fledged larva later onwards, okay? This is larva 1 and this is larva 2. You know what happens? When there is no nucleus, there is no cleavage. When there is a transfer from nucleus, that means the nuclear material at 16 cell stage, then again they become cleavage active. Somehow at the end, he got two full-fledged larva 1 and larva 2 in one experiment. Okay. In another experiment, he has done a separate pattern. Say this is the same new egg. He did not cut the portion not through gray crescent but parallel to the gray crescent. So imagine this is the gray crescent, but he has formed a loop-like structure a parallel to the gray crescent. Therefore, the thing is, what happens? One particular embryo contains gray crescent, but the other embryo does not form. That X have gray crescent and Y has no gray crescent. This is very important. X has gray crescent because he know the baby hair, that means noose didn't pass through the gray crescent, but the noose passed through that means parallel to the gray crescent. So that means so that one part of the embryo have gray crescent, other part did not have. What happens next? part having the grey crescent develop into a full-fledged larva. The other part become unorganized belly mass. Unorganized belly mass. Because it doesn't have axial ends. So it is clear that grey crescent is indispensable for proper embryonic development. Why? How we can come to a conclusion? Because here second part of the embryo doesn't have grey crescent, become an unorganized mass, cellular mass, what we call belly mass. Though it have material for liver, lungs, but it did not become a proper larva. That's like that part which have grey crescent becomes a full-fledged larva, but here in this case it didn't form. So it is a clear-cut example that grey crescent here is indispensable for embryonic development. This is very important. That's how he has performed. That means he has come to a conclusion that there is a certain section of the embryo which acts as an induction, and that means this influence the other neighboring regions of the cell so that they can differentiate into some specific organs. And likewise, he has performed different experiments. Experiment 3, 4, but on the whole, we are not going into the details. Okay, but the inference I am saying, what he has concluded is the dorsal lip of the blastopore. This is very important. The dorsal lip of the blastopore. Dorsal lip of blastopore serve as primary organizer. Primary organizer. This is very important. We have discussed in in gas relation. Say for example, this is a gas law, and this is the lip of blastopore. The dorsal lip of the blastopore is the primary organizer. What exactly the primary organizer? The organizer. Organizer means it organizes the whole embryo. Is very important. What exactly the word organizer? Organizing something. That means just like we are organizing a function, organizing a seminar, organizing the embryo development. What is primary? Organizing the whole embryo. So here dorsal lip of the blastopore is the primary organizer. Because how and what makes it so special? Because it induces gastrulation. Without gastrulation, we have already discussed the crux point for segmentation of the organogenesis is through gastrulation only, because they form primary germ, and these primary germ layers which are formed through gastrulation are under the influence of this dorsal lip of blastopore. This dorsal lip of blastopore induces the gastrulation, and they also form entire dorsal axis, and they also form first one is gastrulation, another one is dorsal axis, which contain notochord, neural tube, and somites. This is very important. This is the first one, this is the second one. Why it is considered as primary organism? Because it formed through, there are certain essential features. It induces the gastrulation, which is a basic platform for rest of the organogenesis. So that means basic platform for the primordial organs, say chordum, mesoderm. They in turn form different organs like forebrain, midbrain, hindbrain, like that. That means neural tube. That means say optic plaque. So these are all depends on primary organisms, what we call dorsal lip of the blastopore. And they also form dorsal axis. This is very important. Entire dorsal axis. That means it consists of notochord, neural tube, and somites. That means chief architect is the basic character of dorsal lip of blastopore, which actually the primary organism, what we call self-organization, and at the same time self-differentiation. That means self-organization means here it has the ability of chief architect. That means has such certain mission to form an complete embryo, and this property is attributed to dorsal lip of blastopore. So in examination they might ask you what is name the part of the early embryo which acts as a primary organism. That is the dorsal lip of the blastopore. This is very important. And these blastopore, they have self-differentiation ability. That means apart from organizing, self-differentiation means not only they induces others, So I have already discussed. They induce the neighboring cells so that they can differentiate different organs like neural tube, for example. And it in turn may act as inducer. So that what we call sequential induction. We'll discuss later. Okay. That means not only just self-organization but also their self-differentiation. They also become part of that embryo. That means an organ of the embryo. So self-organization, self-differentiation is our most important characters to become primary organism. Self-differentiation is the most important. And why it is considered as the primary organism? We have discussed already. Influence the entire embryo. They induces the gastrulation. They induces the formation of entire dorsal axis like neural tube, notochord, somites. This is very important point. That is why dorsal lip of the blastocyst is the most important. That means it is a primary organism and most important as an essential part in embryonic development. That means the entire concept of embryonic induction has been attributed to the primary. That means dorsal lip of blastocyst. Later on, different organs are formed and they interact as inducers. Likewise, it will continue. Okay. We'll just take a particular example how this process ends. Okay. That means what we have discussed now, sequential induction. Sequential induction. We will discuss about the most important concept here. Referring to a particular example, sequential induction. Say I as an example. We have discussed already. One induces others. So there are different organizers. It implied that say primary organizer, secondary organizer, tertiary organizer, quaternary organizer. So primary organizer gives us to one particular tissue. Then it acts as an inducer again. That means inductor, secondary inductor. Then tertiary, quaternary likewise. That means it's all lead to a progressive organogenesis. That means progressive embryonic development, specifically called determinative based embryonic development, because they are determining the kind of organ. Say for example, this has to become neural tube, this has to become notochord, like that. That means determinative basis or mechanism of embryonic development is a crux point for embryonic induction, but in a sequence, not just one step, not just one stop express. It's like passenger train, multiple platforms and multiple stops. It's like cascading effect. Say one particular example we'll take now. Say I, how this forms. We knew that, chorda mesoderm we had also discussed already. Chorda mesoderm is considered as the roof of the archenteron. Primordial gut is the archenteron. Primordial gut is the archenteron. And later onwards, they induce other neighboring cells. How I will tell you. Okay. This is what we call, one particular layer of embryon development. Until how it forms. Something like this. Okay. Then this is another zone. How do you know it ends? Okay. This is the lens placode. This is the lens placode. And this is the diencephalon. This is the optic vesicle. The optic vesicle. Okay. I will tell you how that means lens has been formed in a sequential induction. Say for instance, there will be a chorda mesoderm. which is what we call roof of Archentilon. Roof of Archentilon, which is a primary organizer. Primary organizer. And this primary organizer, they induces a specific organ, what we call, say neural tube. Neural tube. That means differentiation into neural tube into forebrain, midbrain, and hindbrain. And optic placode. Optic placode. These are important. Cardamom system, which is the primary organizer, they differentiate one particular organ into, that means cells into neural tube, into forebrain, midbrain, and hindbrain, and optical placode. And what happens? Say for example, forebrain. That means they differentiate in primary organizer cardamom system, act as inductor, so that a particular cell or a group of cells they differentiate into forebrain and optic area. And optic area in the anterior part of the embryo. This is the anterior part of the embryo. Then this concentrate on the optic area. This optic area undergo evagination. To form optic vesicle. And this optic vesicle undergo invagination to form a double layered optic cup. And this optic cup acts as secondary organizer to form lens. And this lens acts as tertiary organizer to form cornea. That means, say, the primary organizer is cardamom. It induces to differentiate neural tube, say forebrain, and optic area. And this optic area evaginates to form optic vesicle, and this optic vesicle invaginates to form a double-layered cup, what we call optic cup. And this optic cup act as secondary organizer. That means they in turn act as induction source, so that it can induce us to form a lens. How? Outer laying epidermis. That's it. They become lens. And this lens act as a tertiary inducer, that means tertiary organizer, to form cornea. So we have discussed the sequential induction. How cardamom that leads to ultimately formation of cornea. The primary organizer, then the second organizer optical cup, the tertiary organizer lens, and cornea. That means eye lens are the tertiary organizers. They form cornea. And how they form? That means they in turn form from induction force of secondary organizer, what we call optical cup. And how they form? Optical cup, by the overlying epidermis, the ectodermal layer, it converts into optical lens. And optical lens inform cornea, what we call sequential induction. Say for instance how it appears. Say this is we discussed the optical cup. This is optical cup. This is optical cup. This part I am magnifying. Okay, later how it forms. Optical cup. Then optical lens. There is a lens, the optical cup. That means lens vesicle. This is the optical cup. So double layered optical cup, they form optical, that means lens vesicle or optical lens through overlaying, outer laying, what we call outer laying ectoderm. This become into lens. So optical cup is a secondary organizer, and lens in turn act as tertiary organizer to form cornea. This is how this become this. These very important point. That means we have already discussed X is in a cascading manner. X influence Y, Y influence Z, Z A, B. So that means two different respective organs or a specific organ. They're all sequential induction. Say for instance, we have discussed already. Primary organ is a quarter mesoderm. Likewise, forebrain is also a secondary organizer. Hindbrain is also a secondary organizer. Nasal placode coming from forebrain. Forebrain. Say for example, for better understanding. Forebrain. Just giving examples. Certain examples I am giving. Forebrain. Secondary organizer. To form nasal placode. Hindbrain is also a secondary organizer to form posterior part of the pituitary gland. Posterior part of pituitary gland. This is very much. That means we all discussed secondary, tertiary like that. And also notochord, which act as tertiary inductor and quaternary organizer to form temporal membrane. This is very important. That means each and every organ may act as an inductor so that it can influence the neighboring